And this video is on Chapter 15, Part A. Please remember that Chapter 15 will be divided into 3 videos and not the standard 2. So where do we begin? Let's begin by looking at a general structure of a differentiated cell. Now in this cell we have the plasma membrane, which goes around the outside, and then we have much more in terms of internal membrane. So we have the nuclear membrane here, which is a double membrane. We have the membrane wrapping the mitochondrion, which is also a double membrane. And if this was a plant cell, which it's not, it would also have chloroplasts, which have a double membrane. So those are the 3 structures inside a cell that have at least 2 membranes. Remember that chloroplasts have a 3rd internal membrane. But there's a lot more in terms of membrane here than can be resolved by just those few structures. So the endoplasmic reticulum, which is here, has itself a single layer of membrane. So there's the Golgi complex illustrated in green, and a number of other organelles, including these granules, which contain mucus. So these are mucus cells that line our stomachs and our digestive tracts, releasing mucus to protect the lining of that particular organ from digestive or harmful entities outside. So cells can be specialized with various amounts of membrane depending on their particular specialization. The learning objectives for Chapter 15 are quite diverse and varied. And over the course of the 3 videos, we will discuss each of these 15 different objectives in subsequent detail. So let's divide the information into modules. So the first module will just talk about other structures which are bound by membranes that are normally found inside eukaryotic cells. Remember that the vast majority of prokaryotic cells only have their plasma membrane, and there is no internal membrane whatsoever. There are some exceptions. Some photosynthetic bacteria and some specialized bacteria have internal membranes, which are basically infoldings of the plasma membrane. But let's not dwell on that fact. So in Module 1, you will need to know the identity of the different organelles that are enclosed by their own particular membranes. You need to know the structure of those organelles, the function of the organelles, and also the number of organelles in a typical cell. So let's get started. Here is an electron micrograph of a cell, and you can see that the cytosol is occupying a very small fraction of the entire volume of this particular cell. So the nucleus here takes up a large proportion of space itself, and it's surrounded, as we said earlier, by a double membrane. But then packed into the cytoplasm are other organelles taking up the vast majority of that space. So here's a mitochondrion, and here's your rough endoplasmic reticulum. There's some more over here. Then you have these granules in various places, and then you have lysosomes, peroxisomes, and many other structures, as we'll see in the next slide. So how much do the students need to know? If you can remember each one of these terms, what they do, and their structure, and the number that are present in a typical cell, then you're in good stead. The 2 figures are equivalent. They're basically similar. So this is an animal cell with a microvilli. This is also an animal cell with a slightly different arrangement of components. But the labels are the most important thing. So you can see we have the nucleus and we have the plasma membrane. So the nuclear membrane will be right there, and the plasma membrane will be right there. Now between those 2 structures, there are various other membranes that we need to understand. The most common ones will be the Golgi and the endoplasmic reticulum, because all cells have those 2 structures. All eukaryotic cells have those 2 structures. In addition, all cells have mitochondria. All eukaryotic cells have mitochondria. So those are the three most common membrane-enclosed organelles that every eukaryotic cell has. So mitochondria, Golgi, and endoplasmic reticulum. Now the endoplasmic reticulum comes in two varieties, as we'll find out the reason why later on, but just remember that please. Now what students tend to forget are some of the other structures that are surrounded by organelles within the cytoplasm. So we have the endosomes, and the endosomes, as we'll see later on, are simply vesicles that are pinching off, as you can see here, from the plasma membrane and entering into the cytoplasm. And that's what they're called on their journey inwards, endosomes. And they can fuse with other structures inside the cell, or they may stay independent. Other structures are the lysosomes. Lysosomes are present for degrading old biology. For instance, if mitochondria become old, they can fuse with a lysosome and be degraded in that manner. Also present are peroxisomes. Peroxisomes, as the name suggests, contain the ability to produce hydrogen peroxide, and then that hydrogen peroxide is used for degrading and eliminating threats to the cell. And then finally, we have one last reminder that ribosomes themselves are not membrane enclosed. So these are membrane-free organelles. So that's the exception on this particular slide. Table 15.1 from your textbook provides a quick reminder of the main functions of these organelles. Some of them have accessory functions which are listed in the text for this section of the chapter. So make sure you read that please, because you could be tested beyond the labeling that's present just here. But this is something to get started with. And then finally, we need to know roughly how many of each type of organelle are present in each eukaryotic cell. So the textbook authors have taken a liver cell called a hepatocyte and used that as a prototypical example. It doesn't mean that all cells will have the same level of membrane organelles, but this gives you a baseline. So let's start with the most obvious. The organelle that's present in one copy per cell is the nucleus. There's one nucleus per cell, and there's no exception to that. The Golgi apparatus, even though it's a convoluted series of discs-like membrane structures, they are interconnected into a single contiguous membrane enclosed organelle. So there's normally one Golgi per cell. And likewise, the endoplasmic reticulum, even though it can be extensive, occupying up to 90% of the cell's cytoplasmic volume, it is a single interconnected network of canals. So the number there is still 1. More importantly, the volume occupied by these structures is given here. So the nucleus for most people occupies most of the volume of the cell other than the cytoplasm. But as you can see here, it only occupies about 6% of the volume of that typical liver cell. So the remaining 94% has to be allocated amongst all the other structures. The interesting thing is that more than half the volume of the cell is the cytosol, the area outside of the organelles. So that's an important understanding to take away. Focusing our attention now on organelles that are present in multiple copies, we see that endosomes, lysosomes, and peroxisomes are present in a typical hepatocyte in the couple of hundred number range. So these are scattered all over various parts of the cytoplasm. And the number of mitochondria is huge, 1700, because this is a very active cell and it's generating lots of products, so it needs a lot of energy. So students may think a typical cell has a few mitochondria, but they would be mistaken. 1700 is a huge number. So students should pause here and see if they can relate to this learning objective. Moving forward, let's move on to module number 2. Module number 2 is straightforward. It considers why we got to where we are today. So it's looking at the evolution of the endomembrane system. And a typical learning outcome here would be for you to be able to describe how the endomembrane system might have evolved in eukaryotic cells. So let's look at one of two possibilities. So the first thing we want you to consider is how the nucleus formed. What's the reason for the nucleus to form? So we know that modern day prokaryotes, they resemble this structure here. So their DNA is held in place by a stitch to the inner surface of their plasma membrane. And they also have membrane-bound ribosomes, which are able to secrete their products into the extracellular space out here, or to embed those proteins into their membranes. But how did we get to modern day eukaryotic cells? Well, one theory is as the cells became larger, the membrane was not sufficient to support the biochemistry. There wasn't enough surface area to satisfy the needs of the volume of that cell. So that's what we have as the surface area to volume ratio. And if you recall in Bio 2, we spent a considerable amount of time looking at this conundrum of the surface area to volume ratio. And you can see why, because it helps us understand situations such as this. So as the cells became larger and they had that issue of not having enough membrane to support the needs of the cell, one solution was to increase the area of the membrane by folding it inwards. So having these infolds or these invaginations increases the surface area upon which biochemistry can take place. And it's thought that one such invagination made the DNA attached part of the membrane form a cup. And that cup continued to form through mutations until the cup completely surrounded the DNA. And that may have been the origin of a nucleus. And theoretically, it seems to work. And that gave us two compartments. It gave us the brown compartment, which is now called the nucleus, and it gave us the gray compartment, which is called the cytosol. The major benefit of this type of localization is that biochemical reactions can now take place in the nucleus, which would be independent of the biochemical reactions taking place in the cytosol, thus not interfering with each other. And that allows the cells to become that much more complex. As we unite that theory with the theory of endosymbiosis, we then end up with a scheme that can explain both the origin of the nucleus and the endomembrane system, the ER and the Golgi, as well as the inclusion in the cytosol of mitochondria and chloroplasts. So if you're not familiar with the endosymbiosis theory, please visit chapter 1. Thus, in conclusion, you now have enough knowledge to be able to answer this learning outcome. So moving on to a more relatable topic, let's talk about nuclear pores. Nuclear pores. So the learning objective here for you folks is to be able to describe the structure of the nuclear membranes and the nuclear pore and what is required to move material out of the nucleus and into the nucleus with respect to proteins and also RNA molecules. So let's start at a very high level. So the nucleus is considered an organelle, and it resides one per cell in the vast majority of cases, although remember there are some exceptions. So red blood cells used to have a nucleus during their development, but once they mature, the nucleus has been removed to make more space for carrying cargo. Regardless, a typical nucleus contains a double membrane surrounding the outside, and within the nucleus, within the nucleoplasm, we have DNA, and that DNA is not evenly distributed because in an area called the nucleolus, the DNA is associated with protein activity and gene expression, producing ribosomal and transfer RNA molecules. Focusing our attention on the inner membrane of the nucleus, we find that the inner membrane has a slightly different composition than the outer membrane of a nucleus. The outer membrane resembles more the endoplasmic reticulum. The inner membrane resembles a unique entity which is similar to the nuclear side of that membrane. So the very inside of a nuclear membrane is attached to a network of proteins, and those proteins have multiple functions. But the name of that area is called the nuclear lamina, the nuclear lamina. And the nuclear lamina serves many functions, one of which includes attaching the DNA to its secure location in that region of the nucleus. So the lamina not only allows DNA to be localized to different regions of the nucleus, but it also gives shape to the nucleus itself, allowing it to be manipulated during cell division, as we'll see in subsequent chapters. In addition, students need to be more familiar with the nuclear pores, which are very important when it comes to moving material, especially proteins, in and out of the nucleus. So each nucleus has multiple nuclear pores, which are basically large gaps between the nucleoplasm and the cytoplasm. And one thing to understand is that these holes are not as portrayed here. They're not simply just holes through which material can pass freely, but they are very well constructed in terms of organization. When scientists take sophisticated photographs using electron microscopes to produce these electron micrographs, they find that the nuclear pores are dominated by multiple proteins. Some scientists have calculated that 20 different protein types exist out each and every nuclear pore. And when you look at the inner membrane of a nucleus, you find that they have a very sophisticated organization compared to the outer surface. They have these nuclear pore baskets, the function of which is not important to us at this time. If we turn to a cartoon of typical nuclear pores, it'll help us understand how material is passaged from one side to the other. So, as mentioned recently, the nuclear pore consists of up to 20 different types of proteins arranged in a very particular fashion. So we have these fibers, which are the tails of the polypeptides, extending from the globular structure of the proteins themselves. Now, these fibrils are also present in the nuclear pore. So these brown proteins here, they have polypeptides that extend across the opening of the nuclear pore. And the nuclear basket is also an extension of other proteins, forming this very sophisticated structure across both nuclear membranes. Other scientists have localized more detailed functionality for the organization of a nuclear pore, but we don't need to know this much detail. This slide is just to show you that a lot more is known about a nuclear pore than our textbook wants us to learn. So what do we want you to concentrate on? Other than knowing that the nuclear pore is not a simple opening in the nuclear membrane, it's important for us to realize how material passes from one area to another. In this particular figure, we're going to deliver some cargo. Here it is, this green molecule, which is a protein. We want this protein, which has been manufactured in the cytosol, to be delivered to the nucleoplasm. So how does that protein make its way across the nuclear pore? Well, the first thing to note is that the protein maintains its conformation. It maintains its shape and structure. So there's no need to unfold that protein, as we'll see later with other organelles. The protein has on it a signal, and we'll talk about these signals in more depth shortly. So the protein itself carries a barcode that tells the system that this protein needs to be moved into the nucleus. This barcode is recognized by a nuclear import receptor protein, which binds ionically to the protein that has to be transferred to the other side. And then it carries that protein through the nuclear pore by interacting with the nuclear fibrils. And the protein bounces around in a random fashion, believe it or not, until eventually it's forced to the other side. And once on the other side, it releases its cargo, and then that protein is recycled through the nuclear pore in a sophisticated manner. So let's take this figure here that you understand how nuclear proteins enter the nucleus. Let's put a bit more detail behind this image. The size and structure of a nuclear pore makes it available for small molecules to enter the nucleus or leave the nucleus by free simple diffusion. So small molecules can enter the nucleus and leave the nucleus by negotiating their own way past these fibrils. However, larger molecules, the size of most nuclear proteins and beyond, have to be chaperoned into the nucleus or out of the nucleus by those receptor proteins. The situation is very similar for RNA molecules, messenger RNA in particular, that has no capacity to make itself move across a nuclear pore, but has to be taken by a nuclear transport receptor. So without nuclear transport receptors and appropriate other quality controls, a messenger RNA molecule cannot leave the nucleus, which will become important to us in subsequent chapters. There is one further level of detail that students must know about transport across nuclear pores. It takes energy. So energy is needed for driving the movement of something. And in this case, the energy is not used by the protein itself because they can vary from cell to cell and even within a single cell. So the system is very generic in that it uses the receptor proteins to tie with energy needs. So in the first quarter here, the binding of the receptor protein to the respective nuclear protein that needs to be transferred takes place. In the second quarter down here, the binding of RAN GTP, RAN is a protein that's commonly used. So this complex here exchanges positions with the protein being translocated across the nuclear pore. And that results in that protein being released. So the protein is picked up here in the cytosol and released here in the nucleoplasm. The exchange of RAN GTP is very important in causing this conformational change that releases this carrier protein. The receptor protein then returns to the cytosol to pick up more cargo, but it first has to dislodge the RAN GDP. And without this step and the use of energy, the cycling of these proteins would not be feasible. Here's a figure that's more annotated to show you exactly how the cycle proceeds. This concludes Module 3. So you should now be in a position to describe this learning outcome. So moving forward to Module 4. Module 4 has a large number of learning outcomes because students need to be able to relay an understanding of what happens in different circumstances. So protein sorting in this chapter is governed by understanding at least these 8 different mechanisms that are required for moving proteins in various situations. But before we do, let's reiterate something that you should already know. That proteins can only be made by ribosomes. And in this particular text, ribosomes are represented in most cases by a structure that looks like this. So this will be a ribosome. Now, why do we have multiple ribosomes attached to this particular piece of messenger RNA? Because that's the most common way in which we find ribosomes translating mRNA into polypeptides. So the polypeptides are these green structures with little dots. And as you can see, this ribosome here will be attaching right now to the beginning of the messenger RNA. And if you take a snapshot of that ribosome in a few seconds, it'll be right here, and it would have added 3 or so amino acid residues to the growing polypeptide chain. And that ribosome will continue to move until it falls off the messenger RNA at a stop codon. When it comes to protein migration between different compartments of a cell, there are 3 known mechanisms that you need to relate to. The first is transport through nuclear pores. And we sort of covered this in the previous module. Without the help of transporter proteins, this is not a possible task. The second mechanism is transport across membranes into organelles. And we're going to focus heavily on this in the next few seconds. And then finally, we're going to look at transport by vesicles to various locations. And that'll be the subject of the next video. So one discovery that fascinated scientists was the presence of small tidbits of information carried by the polypeptide itself. So small sections of the polypeptide, indicated here in red, carried information that told the cell if and where to move that particular polypeptide. And they became known as signal sequences because they signaled to the cell's transport system exactly what the location of that protein should be. These signals are not carried by all polypeptides. Polypeptides made in the cytosol by free ribosomes are delivered at their destination. So they don't need to inform ribosomes or where they should be sent because they're already in the right place. However, proteins that have to be delivered elsewhere need to be informing their ribosomes that their destination is different from the cytosol. And that's where the signal sequence comes into play. Using some very clever experimentation, scientists were able to show the importance of the signal sequence. In especially genetically modified cells, they were able to manipulate the genes themselves so that the signal peptide sequence was either present or not present on the gene for the blue protein, and the same thing for the green protein. So when they deleted the information in the gene itself for making the blue protein, when it was made, didn't have a signal sequence. And therefore the ribosome just dumped it out into the cytosol. Whereas the gene for the green protein was altered to contain the same signal sequence. And when that was being manufactured by the ribosome, the signal sequence was made and it told the ribosome to dump that into the ER. And that's where they found the green protein in these 2 unnatural positions. What do these sequence signals look like? The signal sequences. Well, the answer is that they vary from cell to cell type, and they also can vary between the signal for a particular destination. And as you can see here, it's the amino acids themselves that form the signal sequence. So this one here, lysine, asparagine, glutamine, leucine, these 4 amino acids at the end of a polypeptide result in the retention of the protein in the lumen of the ER, where this particular sequence here, serine, lysine, leucine, would result in the importation of that polypeptide into these organelles called peroxisomes. You're not required to memorize these sequences, just to know that the different destinations are encoded by different signal sequences, and they can be of different lengths. Let us deal with some specific examples now. Let's look at how proteins enter mitochondria and chloroplasts. So we know from previous information that a mitochondrion has 2 membranes and a chloroplast would have 3. The proteins can be localized to any one of these regions that I mentioned here. So proteins could be embedded in the outer membrane. Proteins could be found in the intermembrane space. Proteins could be embedded in the inner membrane, or proteins could be destined for the matrix. Regardless, the mechanism begins by moving the protein into the matrix of the mitochondrium first, and then moving it to its final destination. So proteins are made by the cytosol, but whenever a ribosome manufactures a signal sequence, the signal sequence directs the final destination of that polypeptide to the matrix of the mitochondria. And that's done through interaction using various other protein complexes called the import receptor proteins, the translocator proteins, and of course there are other proteins here that we need to be familiar with. But first, let's concentrate on what happens as the polypeptide is passed from the cytosol to the matrix. The polypeptide signal sequence intercepts with an import receptor protein. And that import receptor protein docks with a protein translocator in the outer membrane of the mitochondrial membrane. And the two proteins then migrate around until they find and dock with a protein translocator, another one, this one is in orange, in the inner membrane. So the two line up, and then proteins are pushed through both simultaneously and delivered into the matrix of the mitochondria. Now, for this to happen, the protein has to be unfolded. So there's only enough space in that gap in the translocator for the protein to go in as its primary sequence. So any secondary sequence is ripped apart. Once the polypeptide enters the matrix of the mitochondrion, it then will fold into its final shape. And in some cases, it may need a chaperone to help achieve that end result. The final thing to note here is that the signal sequence is cleaved off the mature protein because its function has been completed and is no longer needed in this particular scenario. This figure, extracted from another textbook, does a magnificent job of giving us the entire landscape. So if students begin with number 1, they will understand that ribosomes in the cytosol are always responsible for making proteins other than those within mitochondria and chloroplasts. And that these polypeptides can follow different paths depending on their final destination. So the left-hand side is the endomembrane system, and the right-hand side is the organelle system. And you need to become familiar with this by the time you finish reading this chapter. Let us finish off this section of the video by looking at some more detailed information. As mentioned a few slides back, students need to know a deeper level of understanding when it comes to the process of passing proteins across membranes. So in this example here, we're back to the mitochondria and the chloroplasts, and we see that there are a number of colored proteins here. So the green ones represent the ribosome. The brown one is your signal recognition particle. So this is the first time that you're encountering that. Then there's going to be the SRP receptor, this blue one, the dark blue one. Then there's going to be the protein translocator, the light blue one. And then finally, there's one more that you'll see on the next slide. So what's the function of all these? Well, everything is done in sequence. So the signal sequence is first leaving the ribosome. And as it does so, it is quickly recognized by signal recognition particles floating around in the cytosol. And these particles can also recognize the ribosome. So they bind to both the polypeptide and the ribosome, preventing or slowing down further polypeptide synthesis until they dock with the SRP receptor. Now, once docked with the SRP receptor, they then dislodge the SRP, the brown, and then hand over the entire ribosome, including the signal peptide, to the protein translocator. And the protein translocator binds to the signal peptide sequence, the signal sequence, and then pushes in this hairpin manner the growing polypeptide chain into the lumen of the organelle. And this process is dependent on energy, but the energy comes from the hydrolysis within the ribosome using GTP. The final protein that you need to learn is illustrated here in yellow, and that is the signal peptidase. The signal peptidase has the function of cutting off the signal peptide, the signal sequence, freeing up the rest of the polypeptide. So by cutting there, the signal peptide is cleaved, releasing the polypeptide once it's finished being manufactured into the lumen of the organelle. The signal peptide itself is degraded by enzymes not necessary for us to learn, and then those amino acids are recycled. And this cycle continues as long as ribosomes are delivering their cargo to the organelle. Please become familiar with the previous slide and this slide before moving forward. One of the most important things you can learn is the orientation of the polypeptide. Which end of the molecule is the ammonia end, and which end of the molecule is the acid end, the amino and the carboxyl. Please remember that, because that's going to make a difference as we look through subsequent figures in this chapter. The level of understanding required of students extends to being able to relay how different ends of a polypeptide are positioned in different compartments of a cell. So the orientation of this molecule. In this example, the ammonia, the amino end is in the lumen and the acid end is in the cytosol, as well as how many times the protein passes through the membrane. So this is a single pass of the protein called a one pass or a single pass protein. And sometimes we have more than one pass and the orientation may be reversed. So keep an eye on both the red and the orange segments in the polypeptides. So what's new here? Well, for the first time we're seeing this orange segment. And the orange segment, which is also part of the polypeptide chain, is a stop transfer sequence, and it communicates with the translocator. So the red sequence is required to attach to the translocator and orientate the polypeptide chain in the right direction. But the stop transfer sequence prevents any further passage of the growing polypeptide past the translocator. So it's basically a sticky element. It sticks itself to the translocator. Therefore, this loop of the polypeptide is extending into the lumen, and the rest of the polypeptide will be manufactured by the ribosome out here in the cytosol. And once the signal peptidase has done its job, the protein will be released in this fashion. So we can see the ammonia end and the carboxyl end sitting in the membrane in that orientation. Thus, here we have a single pass protein with this amino end facing the lumen of the organelle. In the second example here, we see we again have a red sequence, but now it's no longer at the extreme end of the molecule. It's been moved further back by evolution. And then there is also an orange segment which has been moved further back too. So in this case, what happens is that this part of the polypeptide is made normally by the ribosome. And when it gets to this region here, the ribosome stops making polypeptide. It's then moved with its polypeptide to a translocator channel on the right organelle. Depending on this sequence, it'll come to the mitochondria. A different sequence may take it to a chloroplast, and a different sequence may even take it to a peroxisome or some of the other organelles inside the cell. But you can see what's happening here. So the red sequence this time sticks the polypeptide into the hairpin structure, therefore excluding the bottom part and keeping the amino end into the cytosol. This will continue until the orange part sticks that part of the polypeptide also in the translocator. So this loop is generated. Now there is no peptidase to remove the red molecule because it's not necessary and there's no signal to do so. Therefore, there's no cutting taking place, and once the polypeptide has been finished by the ribosome, it's released from the translocator as a 2-pass protein. So it passes through the membrane twice. And now you can see both ends of the polypeptide are facing the cytosol, and the loop is in the lumen. Just to round things off with previous knowledge, we know that proteins can be modified at the ER or Golgi level, and those modifications normally take place on the lumen side. Illustrates how glucose entities can be attached to the polypeptide chain to make it into a glycoprotein. Thank you.